Good evening, all. In last lecture, we discussed about SDS page, right? Sodium sulfate that is used as a denaturing agent in protein analysis and polyacrylamide gel electrophoresis of proteins. That is what introduction we had done, right? So here is the summary note of whatever we discussed in the last lecture. Fine. So what is SDS? How it works? Then what are the activating agents when we talk about polyacrylamide gel? How the polyacrylamide gel is prepared? What are the catalysts used? And how is polymerization of acrylamide takes place? That is all we had discussed. So this is the first part wherein in the summary of polyacrylamide gel preparation is uh, just summarized, right? So uh, in addition to this, we had discussed about the SDS page, right? Now here in case of SDS page, if you remember, we had talked about the solubilizers, right? Now, when I say this, what I mean by it, let's look into the summary, right? SDS page, right? Are you able to see this particular slide, right? Now, this is the uh, screen that is available on internet. It is produced by Thermo Fisher Scientific. I'm sharing their screen. Now, if you are able to see the gel, what do you see in the gel? You see darkly stained bands are seen, right? Now here, if you read the information, so monomer, dimer, these are the words that you see, right? Trimer, tetramer, pentamer, and higher oligomers. So here the concern is of oligomeric proteins. So oligomeric proteins are those which are going to have more than one polypeptide, right? So they are referred to as oligomeric proteins. So the whole structure, when you talk about such oligomeric proteins, is stabilized by disulfide bonds and hydrogen bonds, right? So here I would like to share that structure for you. Yeah. So this is something how it would be uh, uh, the important functional groups and the interaction, which is going to introduce hydrogen bonds, then disulfide linkages or hydrophobic interaction within a polypeptide or between the polypeptides, right? Here I am showing a single polypeptide which is folded. And how are the disulfide bridges formed? How is the uh, hydrophobic interaction occurring between the hydrophobic groups, CH2 groups and CH3 groups? And here it is how the hydrogen bond is formed, right? So this is how the case when you are talking about a protein having a single polypeptide. Against that, here I'm talking about a polypeptide, not one, but multiple polypeptides are there and molecule is oligomeric. Now, when you have to do the protein analysis of such oligomeric protein, the whole structure, as I said earlier, is stabilized by hydrogen bonds, disulfide bridges, or maybe the hydrophilic interactions, right? So these proteins, if they are going to migrate during gel electrophoresis, how it would look like. So for that purpose, I have shared this slide. So if the subunits of these proteins are not separated, so the protein will migrate as a single band during gel electrophoresis. It will form just single band if it is not solubilized. So that means the polypeptide chains are not separated. But if they are separated, then each single band will be produced by each subunit, or each polypeptide will form one separate band. And that is what we are seeing here in this slide. So the subunit of the proteins are separated from each other, and then they are allowed to migrate. Now, when they are to be separated, how are they separated? How is the quaternary structure of protein destabilized? When I say destabilized, that means the native structure, the functional conformation is destabilized, right? So the chemicals or compounds or rather I should say substances and that are used for destabilizing the quaternary structure of protein are collectively called as solubilizers. The word itself tells you solubilizing, right? So for example, You take an example of say a protein, say RNA polymerase. Okay, when I say RNA polymerase, it has got three subunits, alpha, beta, and beta prime. Now they will migrate as a single species if subjected to zone electrophoresis. But if they are treated with SDS, the solubilizing agents and then electroforest, so they will form three different bands, right? Now here also you are saying if it's a monomer, it will form a band here, but if they are dimers, you are seeing bands, right? So depending upon the number of monomers present in a given gel, you can find the bands. If the monomers are not separated, a single band will be produced, and if their monomers are destabilized by use of say urea, so generally urea 3 to 12 molar solution of urea is used, that will disturb the hydrogen bonds, right? And macromolecular complexes or aggregates, the structure of which is solely maintained by hydrogen bonds, will dissociate. So urea is a solubilizing agent, right? So urea in the gel in which electrophoresis is conducted is important now. So you have added urea in the gel. So that is the advanced version of it is referred to as denaturative gel gradient gel electrophoresis. That is for DNA also you have urea added to the gel and then it is analyzed. But here I'm talking about proteins. So when you have added urea to the gel, then additional information about the macromolecular structure, say, of the proteins, right, that can be obtained. So many proteins are studied with the help of urea as a destabilizing agent, and then the protein is electrophoresed. So macromolecular structure like myeloma, gamma globulin, light chain, heavy chain, then haptoglobulins, then alpha beta caseins, say E. coli ribosomal proteins, etc. Are studied using this urea as a destabilizing agent, and then electrophoresis done. And as I said earlier, double stranded DNA also can be rendered single stranded when you add urea to it, and then on the basis of uh, that you are going to go for DGGE technique, that is the gradient gel where you are using denaturing agent. So denaturing agent, gradient gel electrophoresis, that is used in NGS technology, that is next generation sequencing in DNA. Of course it is advanced version, but just because it is basically the same ingredients and same procedure, I refer to it. Second one is SDS, sodium dodecyl sulfate, about which we have discussed in the previous lecture. Mercaptoethanol is another one which have are used to break the disulfide bridges, right? So mercaptoethanol and SDS together, we have discussed how it is utilized for the process, right? So maybe you are going to have proteins that are insoluble in concentrated aqueous solution of urea. So when you have to do electrophoresis, it will be a question then, right? So in that case, you have to take help of say different type of solvents may be added to it. So maybe phenol, acetic acid, water, that is a common composition. Phenol, acetic acid, and water in 2:1 proportion is used, and this solvent system is then going to be used for separation of such proteins. Example, where phenol, water, and acetic acid as a solvent system in addition to urea is used is ribosome subunits. So you know ribosomes are made up of different subunits. So 16S, 5S, 5.8S, etc., etc. That is what you talk about, right? So those proteins can be so these are the RNAs, 5.8S RNA, 16S RNA, and along with that you have proteins associated. So those proteins can be separated using the solvent system in addition to urea, so phenol, acetic acid, and water. So in nutshell to say, whenever you have oligomeric proteins, that time you have to separate those polypeptides monomers, and then you have to run the electrophoresis so that you can reveal the true and complete nature of the molecule. Otherwise, even though there are so many polypeptides, a single band will be produced. So here in this case, if you talk about characterization of beta 2M oligomers. right? So SDS page of that particular protein is done under non-reducing and reducing conditions. So oligomerization started from monomer. This is what, right? With or without DTT. So that is an agent which is added to it, and then SDS page is done. So DTT is added, agitated at 37 degree for various times, and after 5 days without DTT, oligomer ladders, they are observed, right? So of course this protocol you need not remember. It is just to make you understand the concept how oligomeric proteins would get separated using SDS page. Fine. And that is how you can have monomers to higher oligomers separated. Now here the another term that you are seeing is the stacking gel. This is what we discussed in the discussion while we were talking about the intro in the introductory lecture about electrophoresis, page electrophoresis. So what is the stacking gel? How it is going to function, that we have discussed. I'll reconnect you with that. The stacking gels are the pre-gels, you can say. This is the dissolving gel, and here 1 centimeter area as against this is 10 centimeter gel. If you consider, 1/10 of it is stacking gel. So stacking gel is the one which is used for concentrating the protein in the particular gel, because for example here we had talked about the percentages of various materials that are used and the role of the stacking gel. I'm trying to reconnect you to that. Let's check. Let me check if it is available here immediately, or else uh, these are the previous ones. Just give me a second. Okay, fine. So yeah, this one. Here you are able to see what I'm talking about is as stacking gel and the resolving gel. This is the resolving gel and this is the stacking gel. Here the compositions are little different. The buffer system also different. We have talked about it in details in the previous discussion, right? So when we are talking about the stacking gel, the role is to concentrate the protein under analysis and then it will be resolved. That is, bands will be separated in the resolving gel. So, stacking gel preparation is one of the things that we have discussed earlier in previous lectures. Now, here we can look into it again in little of details, right? So, how you are going to load the sample, how the electrophoresis run is going to occur, and how you are going to get separation. So if you are able to see here, it is the buffer tank, right? Then here also you are seeing that the wells are covered and this is the buffer in it, right? And here are the separated bands, they are seen, and the bands are stained with Komasi blue staining. So these are the three steps, you can say. Protein sample are loaded, the direction of migration you are seeing, so it is allowed to run, and then it is visualized. All right. So for that purpose, what you need are the different chemicals. The very first thing is the gel matrix. How to prepare gel, we have discussed in the previous lecture two details, including ionic strength, pH, concentration of acrylamide, bisacrylamide, etc. And here is the summary of that, just to reconnect. So acrylamide, bisacrylamide solution, when you are talking about, they are taken in specific concentrations so that polymerization would occur and the pore size can be governed. Then you have gel casting buffer. So when you are talking about gel casting buffer, generally it is tris HCl, pH at 0.8 is used, and you have to prepare that. So maybe you are taking 18.15 gram of trees base in 80 ml of distilled water. You are adjusting the pH to 8.8 using HCl, and then you have to make up the final volume. The procedure also is given in the slide. Right. Now, when you are preparing the stacking gel, so here you will get to know that the buffer that is used for stacking gel and the buffer that is used for dissolving gel also are different. So 0.5 molar is tris-HCl, pH 6.8. And when you are talking about resolving gel, the buffer is 1.5 molar tris-HCl. See, the molarity of the tris-HCl also is different, and the pH at which it is going to work also is different. So resolving is at 8.8 and the stacking gel is at 6.8, right? Because the purpose of stacking gel is to concentrate the sample in the stacking gel, so it will run over the dissolving gel perfectly. And of course, when we talk about the loading buffer, the composition includes bromophenol blue, mercaptoethanol, glycerol, SDS, and tris HCl. So, when we talk about the equipment, it consists of two components, as you have seen. Here, buffer is shown, but before buffer reservoir is there, through which it is going to be put into the electrophoretic system. And another is the power supply. So, these are the two things. So buffer reservoir has upper and lower buffer reservoir connectors there, and they are connected by gel. So at one end you have buffer reservoir, another end you have a buffer reservoir, in between you have the gel. So platinum electrodes are there, which are going to be the power supply providers. Current will be conducted through those. And once you have prepared the high density component, right, where you will have to prepare the high density component, of course it is in the stacking gel. You have to load the sample, right? So it should be of high density, so concentration of the sample would be taking place. So to that you can add ficol or you can add glycerol. And of course this will help to prevent mixing with the upper reservoir buffer is loaded on the top of the gel, the another function of it, right? So when you are preparing the sample, it is prepared in a high density component like glycerol or ficol. And what is the benefit of that? It will prevent its mixing with the upper reservoir buffer. Otherwise it will get distributed in the buffer and it will not be available for electrophoresis process to occur. So entire sample has gone into the buffer, so you won't get separation of the analytes. So you are adding a density component, right? Now you have to load the sample, and of course it has to have a tracking dye, and that is nothing but the bromophenol blue. Generally very low concentration, say about 0.004% is utilized when you are talking about the tracking buffer. So you have glycerol or ficol, you have sample, you have tracking dye that is mixed, and you are going to observe the electrophoresis front. So this tracking dye bromophenol blue is going to migrate, and it is going to be like a kind of indicator, or it will show us for electrophoretic process. So you can say that extent of migration of the dye will tell you about how far electrophoretic process has happened. Because the dye is a smaller molecule, it migrates faster than all other molecules, right? So if I have to see the structure of dye, this is the bromophenol dye molecule, right? So sorry, this is comaciblen blue, it is for staining of the protein, I'm sorry. I thought I have added that structure of bromophenol blue also. I'm sorry. I get back to the point. So tracking dye is bromophenol blue, and it is smaller molecule. It is going to be smaller as compared to the protein under study, right? So it will move fast. So it will be moving faster than the macromolecules and will be indexing the electrophoretic process. Thus, if the electrophoretic process is stopped before or just after the dye comes to the bottom of the gel, otherwise it will run in the other direction. So it will tell you about where to stop, right? Generally when you talk about pH, it is fixed at 9, which gives net negative charge to most of the macromolecules, and the anode to which these negatively charged macromolecules will migrate under applied electric field. So the pH also is, as I said, 8.8 to 9 is that of the dissolving buffer. So when the power is switched on, you are going to see or observe or monitor the running front, and then when the running front, that is indicated by bromophenol blue, goes to the second end of the gel, then you are going to uh, stop the process, right? Now, of course, when you are talking about these electrophoretic processes, you have submerged electrophoresis, you have advancement of it. Those advancements like electrofocusing and all, we will talk in the next lectures. All right. But now to look into the polyacrylamide gel that we have talked, you are going to have separation of the bands, and they are going to be stained with the staining agent. And let's look into that, how to stain the bands. So here This diagram is showing you how you are going to load, how you are going to run, and you have to visualize. So how to visualize, that is the next part in the discussion. Now here you have to fix this gel. So gel fixation, because once it has run, even if you remove the electric current, diffusion is to certain extent might happen because it is the porous structure and it is in the liquid medium, mobile phase is there. So diffusion on account of mobility in the mobile phase would spread the bands, right? That should not happen. So you have to fix the gel and then you have to stain the gel. So when we are talking about sorry gel fixation, generally methanol, glacial acetic acid and water is the system that is used. And when it comes to staining, there are different methods of staining, but commonly used are Comasi brilliant blue staining and silver staining, right? So in the previous structure you have seen the blue color bands, and here you are seeing black color bands. The black color bands are on account of silver staining, right? So you are going to use various types of stains, selectivity, or maybe mixed staining is also there. Staining with fluorescent dyes also is one of the techniques that can be used, right? Now here is the general protocol of whatever I talked till now. So you are going to have a stacking gel, you are going to have a dissolving gel. The buffer composition for both the gels are going to be specific. Loading of the sample we have talked, right? And then when you are going to allow the movement, the proteins will migrate. Here, this is the electrophoresis process, right? But now before that, I'm talking about the pre-procedure also here, how you are going to insert the combs and how you are going to cool the solution, how you are going to pour it. So it is general protocol, you can say, of the process. And after staining, you have to hold the stain, then you have to de-stain it, and then you have to observe the gel. That is how the steps are, right? So staining step, you have to wash the gel several times, so excess stain is to be removed. So of course, you can use the destaining solution like methanol, right? Can be added to it for destaining, and excessive stain can be removed. And then you are going to take the photograph of it. That means you are going to gel dog the system. Now, when I talk about silver staining, you are going to immerse the gel into the fixing solution for 40 minutes, or an overnight immersion in the fixer also is suggested. But of course, fixation time will depend on the concentration of the fixative solution that you are using. All right. So from the fixing solution, you are going to remove it, wash the gel for 10 minutes with 30% ethanol solution, and followed by a wash with ultra pure water. So overall, you have to put it in the staining fixative solution first, then staining solution, then wash it, and then you are going to incubate the gel in a sensitizer solution for 10 minutes. Now the sensitizer solution also is removed, and then you are going to put it in the silver solution. Then for silver solution for 1.5 minutes to 2 minutes, it is suggested you are going to discard the water, immerse the gel in the developer solution. It is something like your photographic film develops. So the components are developer, fixative, like that, and then you are going to develop a film out of it. So but in this case, you will see black colored spots and not the blue ones. Comasi brilliant blue would give you black colored spots, but this would give you blue. I'm sorry. Comasi brilliant blue will give you blue color spots, but staining with silver will give you black colored spots. Right. Now here I want to share some research paper with you that is telling you about separation of, yes. Just give me a second. Separation of different proteins using gel electrophoresis system. So uh, how you can analyze the components that I wanted to share with you. Just a second. Fine. Until then, yeah, this is the one. Yes, I'm sharing it again. Yeah. So here is the paper that talks about sodium dodecyl sulfate, polyacrylamide gel electrophoresis, and Western blotting analysis via colored stacking gels. So all the three things together. So I chose this paper to discuss with you. Here you are going to have standard SDS page stacking gel and acidic dyes that are used. They are added to the stacking gel. So can you imagine a gel? The resolving part of the gel, the resolving gel part is going to be stained with the tracking gel, and the upper part, the stacking gel, is colored blue or something yellow or orange color, right? So colored gels you are going to have, and they are compared with non-colored gels. So you can say it's a kind of advancement. So here sodium dodecyl sulfate polyacrylamide gel electrophoresis is a standard method we know for protein analysis, and the stacking gels along with the dyes, so maybe tetrazine or brilliant blue FCF or new cocaine. These are the dyes, acidic dyes they have added. So easy visualization of stacking gel also, not only of that of the resolving gel, but the stacking gels also are resolved. So this is a photograph that shows these are the stacking gels in different colors, pink, blue and yellow. So different agents are added to it, and that is how you can monitor the what you say, concentration of, or the protein sample getting concentrated in the stacking gel, and then it will run over the resolving gel and it will migrate and get resolved into components. Right here, uh, the concern is why it is used and who developed this system, right? So in 1970s, SDS page was talked by Lamely et al, right, that we have talked about. But Shager and von Jaguar, they have come up with the new technique because to detect proteins with molecular masses below 20 kilodalton was found to be a challenging thing. So new method they have talked about. So after the proteins are separated by SDS, they are stained using a method that is Kumasi brilliant blue and silver staining. But here, what is this new method that this paper are talking about is tricene SDS page. So tricene is an additional compound to SDS page. So I am talking about advancement in the SDS page also. It is nothing new here, but only the story of SDS page doesn't end with use of mercaptoethanol in SDA, a stage beyond that. So I have taken this paper to discuss with you. Now you can see Western blotting is a kind of application of electrophoretic process, because once you get the bands on the gel, they are transferred onto the filters and then blotting is done further, right? So it is one of the popular techniques for detecting targeted proteins, and you are going to add maybe antibodies, right? And then Western blot can be developed. So this technique was developed in 1979. So this is one of the applications of that. Now here is another technique, that is brilliant blue FCF or cocaine. These are added to SDS page as a tracking gel. So you can see it as a advancement in SDS. The tracking gel is one, stacking gel is another one. So wherein you are going to add different coloring agents or dyes. Now let's see how the process is going to be uh, running, right? So um, I think, yeah. Tetrazine, brilliant blue FCF, and new cocaine, these are used as food dyes, right? So what are these molecules? Why are they used? That is the concern. So 12% acrylamide gel, separating gels are used, and to that they had used these additional dyes, right? So you can have this kind of a gel, right? This kind of a gel in which the starting part is colored with a different dye agent. So it is tricene SDS page, one example of that. right? Is it clear? So that advancement, when I say, it is the one of the advancements. Is it that all? No, there are many more things to be talked about. Let's go for summarizing those. When I say applications, one application, as I discussed, to resolve the smaller sized proteins, you have to concentrate them in the gel. So stacking gel is there, and addition of the dyeing agents, acidic dye agents, they are added. Now here, when I had to summarize the applications of SDS page, protein purification is that isolation and characterization of proteins. You can determine the molecular weight of proteins. You can check the purity of proteins. How you can check the purity of proteins? That if it is run, it should give a single compact band. If there are more than one band or fuzzy band, you are going to say that it is not a pure protein, right? Of course, addition of single metal ion or a ligand also can change what you say, the properties or characteristics of the zones that are formed. We will discuss it in the next lecture, that is how to run the how to study the chromatogram, right? But when it comes to applications, the purity of protein you are talking about, you will see a single band that is formed, right? Similarly, you can find the or identify the variations. So isoforms, you can find, or the strain species, that is from different bacterial species. If you isolate the same protein, is there any variation? Then that can be very well resolved in using SDS page. Then protein-protein interactions can be analyzed using SDS page. Of course, protein contaminants can be detected. Then protein peptide mapping can be done. Post-translational modifications you can visualize, right? Because if post-translational modification, that is once the protein is formed, further maybe carbohydrates are added, glycosylation may be there. So change in the molecular sizes, and so the band forms are going to be varying. Of course, HIV testing and all, where you have the basis for detection of viruses or different proteins from the viruses can be analyzed. Western blotting and protein ubiquitation, that is another application when it comes to SDS page. We have to discuss all of these one by one, but I'm sorry, I have to share something else with you right now. Yeah, because last 1 minute is remaining, so that I cannot start the new topic as such. But yes, here are the applications, and yes, this is another application that I have to talk about, wherein you have, say, proteins which are differing in their molecular size. Very rarely they are differing, right? But there is difference in the charge. So they can be differentiated on the basis of isoelectric focusing. When I say isoelectric focusing, it is based on pi value, that is pH at which the net charge on the analyte is zero. There is no net charge. So can it be used as a principal component and blend it with SDS page electrophoresis? Yes, that is referred to as isoelectric focusing. So it is modification of the method, and here you are going to differentiate on the basis of charge, right? So polymorphism in proteins can be very well detected where they differ by single amino acid, or they differ by the total net charge on it, and which cannot be detected by conventional electrophoresis, can be detected by isoelectric focusing. So separation is based on a pH gradient gel. So here the gel types that we were talking, you have resolving gel, you have stacking gel, a stacking gel that is having colored component. In addition to that, now here I'm talking, one more thing we talked, the gel with the urea, the degenerative gradient gel electrophoresis. So DGG technique, where you are adding urea to it. So similarly here you are adding gradation, but the gradation is on the basis of the pH. So how can you have a gel where the areas in the gel are having different pH? A single gel with different zones having different pH. This can be availed or achieved by addition of ample lights. So we have to talk this technique in details in the next session, maybe today or tomorrow. But isoelectric focusing is possible, and here you can differentiate the protein polymorphs, right? So based on the pi values. So you are going to have the amphibole zones, and wherein the specific protein, when it reaches a zone where its pH, net charge on it, matches with its pi value, and then the protein will have net zero charge, and it will stop at that point, and it will not migrate ahead of it, right? So that is one technique that we have to discuss. Another technique is two-dimensional electrophoresis, which is having a lot of applications in proteomics. Maybe initially you are doing blue gel, that is another technique in electrophoresis. Blue gel electrophoresis, that is also we have to talk as an advancement. It is not there in the syllabus, but at least you should be aware about the terminologies. So blue gel is followed by SDS page. One gel, one dimension, second dimension. These are the concepts that we have to talk. So two-dimensional electrophoresis is another technique which is going to involve both standard electrophoresis and followed by IEF, isoelectric focusing. So separation of bands is on both charge and pi values. So in the first dimension, you are going to run the normal electrophoresis, and in the second, you are going to run it with the help of IF values or pi values. So the 2D spot map you are going to find, and that is proteoform of the gel you are going to analyze. So this is what we have to discuss in detail as applications, right? But just to write one word sentence about the applications, this is the summary slide. So we have talked about separating gels. These are the pre-gels, right? Which are going to have different composition as compared to the resolving gel. Then the electrophoretic process, when we are talking about, we have to take into consideration the buffer glycine component is there, and buffer reservoirs are going to be in two forms as a zwittrions, right? So net charge, say for example you take glycinate anion, it is going to be negatively charged. So role of glycinate, role of chlorides, that also we have talked in the previous lecture. So I'm not going to revise it. But these are the points. I am just touching those for some of the students who have joined now and have no connection with the previous lecture. So role of stacking gel, role of stacking buffer, and the ingredients in that. So chloride ion, glycine, and sample, they are there. So here you are going to have benefit of glycine, you are going to have benefit of chloride ion, as they are going to be running at different speeds, and the protein another depending upon its own speed. So glycinate and chloride are going to act as two discs, and your sample is going to get sandwiched in that, or you can say concentrated in that, and it will set in the stacking gel properly. So these things we have discussed earlier lectures, and in the next lectures we will talk about the gradient gel and isoelectric focusing.